ले लेना फिर हम, ले लेना, नहीं तो फिर मैं मीटिंग में जाऊंगा तो लगाए हुए चला जाऊंगा. Okay, so welcome back after the tea session. So now we will take up few aspects of the gene network. Okay, so gene network, as the name suggests, it is a network of genes which is related to structurally or functionally to the function of the gene, and it has a very important role because it can be detected or it cannot be detected without studying the genome. So if you can see the gene network, template gene network from annotated genome, it is fine, otherwise you have to do the whole genome. So sometimes it is a bit challenging to get the gene network if the whole genome is not available. So the cost of the whole genome is nowadays reducing, so you will have the opportunity to have the whole genome sequence available, then you can connect the this gene network. Without whole genome, you cannot have this data available. A gene network, interaction network is a set of genes also called as nodes connected by edges representing functional relationship among these genes, how these genes are connected together. These edges, namely interactions which are connected, they are also called as interactions, and they are connected through various metabolites, proteins, or any type of functions, or which is affecting the other genes activity. So why it is important to study is that these edges, especially these nodes, these genes are connected for functional connectivity. They are not interconnected, they are connected for giving the functional parameters. Like if you want to express any protein, you want to work on any specific type of metabolite, you want to enhance the metabolite yield, you want to understand the function of that particular gene in any disease model, whatever is there, you use this gene network. So especially this is important in terms of the alteration activity, how these genes are alterating one activity to another, or how they are able to change the function of the gene. So that's why gene network is very important to understand. Besides these physical interactions, there are genetic interactions in which two genes, two variants have a combined effect that does not manifest itself with only one alone of them. So if a single gene is there, that gene will not give the proper interactions. So there are two genes which are connected together and give the function, and sometimes they are not connected also. So it is the place where you get lot of network, lot of genes available, but you have to identify the crucial network. That is the major challenge, and it takes a lot of time to decide what are the specific network involved in the functional analysis. So any case study you take, I will show you one case study here, and how the gene network is to be built, the flow diagram and some basic details about it. But if you are able to build a gene network of any application which you are doing, it gives a very novel output. So people will always remember that you developed this gene network. Okay, so if you are working on any area and you go to the literature, you will not find gene network and you try to build it up. Mostly this is a new technology, gene network, so many people they are not doing it, and the limitation is also the technique. So they are not trained in the technique, so they cannot use it. So we face difficulty because of the technological gap. So with this course, I think I will be able to tell you about something. Now gene network, there are three types. If you see, regulatory, biochemical and signal transduction pathway network, gene ontology and ad hoc networks. So three aspects are there. Mostly you can target all three together also, you can target one also, but our more focus is here, our group. Okay, we are not on this, and this is also not into our domain right now. So regulatory network is main focus for us right now, but this has a lot of role, especially when you talk about the medicine, you talk about other field applications, cancer biology. This plays a very important role, and this also important because it provides the functional information and behavioral pattern in the genes. So regulatory network, they regulate the expression of the genes, and they also give specific information about the genetic makeup of that particular gene, how this gene is designed and how this gene is evolved over a course of time. You might have seen, if you are working on mutant strains and wild type strains of microbes, you might have seen that after 2 years or 3 years, the same mutant strain or some genetical recombinant strain is not able to give the similar type of production. So why it is so? Like my experience with cyanobacteria is that we had one cyanobacteria which was evolved in the lab only through the gene evolution. So after some time you are not getting the same profile or same type of xanthophylls or whatever productions you are looking for, and the specific gene is also not available, the gene is also evolved. So those things are connected and they have to be seen through regulatory network, how this regulation is happening, what is the regulation pattern, what is regulatory network, and how many genes are crucial for regulation of the transcriptional switches. So these things are to be seen through the regulatory network. Biochemical and signal transduction, well defined biochemical, cellular and signal transduction pathway can be considered network as their gene members are interconnected, because these are the pathway. So any one gene will not work or not connected, the whole pathway get distorted or will be unorganized. These network can be regulated at the transcriptional level, at the level of protein-protein, metabolite and protein metabolite-protein interactions. A change in the structure or function of one protein in the biochemical pathway can have overall consequences for pathway functions, possibly even indirect effects on gene transcription through regulatory feedback. As I told you, any single gene can change the whole pathway. So it is like a switches, you see, okay, kahin pe thoda sa gadbad, everything will be. So this is a very crucial aspect. I will show you some gene network, you see complicated structures, okay. Then gene ontology and ad hoc network. It provides functional information about a gene or set of genes associated with behavioral phenotype based on the functional analysis which you have done. Geoterms, gene ontology terms are largely species independent, and they are compared across the species. Geoterms provide convenient higher order classification for genes and proteins that allow comparisons among species, and also specific information of the in the species level at all the level. Now, what are the key components? This is a very simple slide and very easy slide. What are the components in network? Okay, when you talk about network, you have to understand the terminologies. The first term is genes. In gene network, the genes is the fundamental unit which carries the instructions for protein synthesis. There are nodes in a gene network. So the nodes in the gene network are important. We talk about network nodes, okay. Then transcription factors, proteins that bind to DNA and regulate the gene expressions. They are the transcription factors. They influence whether a gene is turned on or turned off. So these are the second terminology, transcription factors. Regulatory interactions, these are the relationship between genes and regulators like TFs, other genes, RNA, which can be positive or negative. If it is positive, it is activated, and if it is negative, this is repressed. So gene is activated, gene is repressed. Then mRNA, this is messenger RNA produced by transcription, which carry genetic information from DNA to ribosome for protein synthesis. So gene network ke liye you need to also have the transcriptomic data. That is also important. So you should have those data available, then only you can build up the gene network. It is not that you do it in silico. You should have those data, and if this data is available in some form in some server, you can also use that data. Then non-coding RNA, they are small regulatory RNA molecules, microRNA. They influence the gene expression at post-transcriptional level. Most of the people working on cancer biology, disease biology, they focus on this aspect. I think this is important, and I think last year Nobel Prize also given in this miRNA. Okay. So you can see that the importance of this gene network. It is not only that application is one-sided, it is multifaceted application. So if you talk about gene network construction, and if you are able to learn this technique, it means you get lot of information about that. Then proteins, the product of gene expression which carry out various cellular functions, signal transduction pathways, communication pathways that relay the signals from the cell's environment to the nucleus, and they influence the gene expression. Then low molecular weight compounds like small molecules, metabolites which which influence the gene expression, and they interact with other network. Significance of the gene interaction network. What is the significance? Why we need to do it? I already explained you various application, but these slides give you a better outcome of that. So helpful for understanding cellular function and regulation. Cellular function regulation, very big term. We have lot of cells in the body, lot of cells doing lot of work, but how they are connected, how they are regulated, how the genes are interacting to the process, how change in one gene will affect the another gene expression, that is all you can study through gene network. If you are not able to have gene network study, you cannot study them. Okay, these things are not possible. One gene you can study, another gene you can study 10 genes, you can study 20 genes, but you cannot study how gene number 2 is helping or having some interaction with the gene 3, gene 4, gene 5, and how this network is built to give the proper function. Second is the modeling complex biological process. Biological processes are always complex, you know, how it is happening. Okay, like again neurogenetic disorders, okay, then you see various diseases which are coming up, okay, and also some of the diseases which are not curable. So there are a lot of problems with those diseases, and there is no understanding that how this disease came. So you have to have this also, that the biological processes which are connected through metabolic diseases, okay, diabetes is also there, okay, and other diseases. Then signal transduction, it can be, and it is helpful in the disease prediction. So this is also one application. Then understanding the development and evolution. They determine how, like geo terms which I have given that orthology. So understanding the development and evolution of specific cell types, tissues, and how different species development is changed or their evolution is related. Applications, you can see drug discovery, potential drug targets you can identify through gene network. Also diagnosis by targeting specific genes and pathways. You can also think about it that okay, specific network you identify, and if this network is available through the marker, like you see biological markers, there is a lot of studies happening on biological markers, but how these markers are connected, how they are expressed, you can see that, and many markers based tests are available in the market. You can see, you take the blood sample, they will tell you okay, so this disease is there, or this disease may come, you have this marker biological, and then marker based discovery comes to the diagnosis, you target that, personalized medicine, okay. So if you see gene regulatory networks differs in individual, the best treatment can be decided according to their specific need. So every individual is different, you know, okay. So the gene network will be also having some changes. So if you want to decide a medicine or personalized medicine, or like you talk about antibiotics, okay, ARG, you have heard about it, antimicrobial resistance. These days the physicians, they will not give medicines without the antibiotic sensitivity test. If they have to give antibiotic, they will do the antibiotic sensitivity test. You might have seen I know that there are a lot of antibiotic, for that we are registered. So our body is not able to, the antibiotic is not able to give effect to that. So they do antibiotic sensitivity test. So when you talk about personalized medicine, that also comes under that. So if you go to any doctor who is treating UTI or some other diseases, you can ask him or her that how you prescribe antibiotics. They will tell you, if you see any prescription, you will see that they are doing antibiotic sensitivity test. Then they will, based on that, they will prescribe the medicines, because they are doing the personalized medicine. This is a personalized medicine. So which antibiotic will be more potent for that particular pathogen, that has to be prescribed. So this is important, and this is connected to that. Then gene therapy, correction of the genetic defects by altering the gene expression. Now tools for gene network finding. This is very interesting. Tutorial iska we will cover up in the next class. Okay, this is very important thing, tools for gene network finding. We started learning gene network since last 1 year only. Okay, so I don't have much knowledge about it, but what my student told me that, sir, this is very interesting but complicated also, because it takes 2 year, 3 year, 4 year, or maybe more than that to understand that concept. Is a new one. So these are the tools. GeneNetwork.org is a free scientific web resource used to study relationship between differences in genes, environmental factors, phenotypes and diseases. All things together. Any application you want, you can use it. String, you know about this software. That is a biological database web resource for predicting the protein-protein interactions, and the string database contains the information from numerous sources, including experimental data and computational prediction method. Then Cytoscape, this is also a very good tool. You know that it is open source software for integrating, visualizing and analyzing the measurement data in the context of network. Then GenMania, it is a flexible, user-friendly web interface for generating hypotheses out of gene function, analyzing gene lists and prioritizing the genes and functional assays. So these are few tools. I think you have this literature available, so you will use this in the tutorial also, some of them. Okay. Now, why networks are important? Though I have explained you, but this gives you a better outlook. Why to do that? Okay. Every time you ask question, why to do that, how to do that, why not, why this, everything you question on yourself. So networks are commonly understood structure to reduce complexity. They are more efficient than tables. So networks to be studied in proper way, because they are efficient, they are easily to understand. Then network tools allow to analyze first, then visualize, both things. So when you do visualize and analyze, it's fine. You do only visualize, don't analyze, no problem. But the problem will come in understanding. So you visualize, you present your result, okay, it's done, but you need to analyze it. So networks will allow you analyze and visualize both. Analyze means characterize the network properties. What is the property of this particular network? 1, 2, 3, 4, 5, 6. I will show you that like this. You see nodes. Okay. I will show you one example also, and also one study which we are doing. I will show you. So how this network is connected to certain metabolite production. Like we target about maybe 4 proteins, for example, those which the proteins are important for the stress of particular cyanobacteria. So we targeted RNC, ribonuclease proteins. Okay. Now how these proteins are involved in the stress genomics, that has to be seen. So for that you need to understand the network of genes which are interconnected for stress signals. So same way you do the network properties characterization, identify hubs and subnets. This is also a big task, identifying hubs and subnets, classify, quantify, correlate, cluster nodes by associated data. What are the cluster nodes? Okay. So it is I think if you see the railway map of India, okay, you see junctions are there, okay, small station, big stations, okay, and junctions. What is junctions? Junctions means from there the train will go to here and there. In the same way here also, if you see railway map, GIAN map, GIAN networking, okay. So then you will understand कि यदि train को if the train has to go to New Jalpaiguri, how it will go, okay. If the train has to go to Shahadra station, how it will go, and if the train has to go to something which is apart from other network area, other junctions, how the train will go. So same way you have to identify, classify, correlate these area, then visualize, explore data overlays, how they are overlaying each other, interpret the mechanism, how a process is modulated or attenuated by stimulus. Then you can see here in the networks, nodes are genes, input is transcription factor, output is gene expression, arrows shows the interaction. Terminology, okay. Nodes jahan nodes bolenge, it means the genes, okay. We use the term nodes, okay. Input means TF, transcription factors, proteins, okay. Output gene expression, arrows means the interactions. So if you see this one, input signal, gene regulatory component, primary output and terminal output. So in this way this scaffold will tell you the importance and significance of networks. Now if you see this, the levels of organization of complex network, the node degree provides the information about the single nodes. The 3 or more nodes present a motive, and larger group of nodes are called as modules or communities. So these are the terminologies which I am explaining. So larger group of genes, okay, or they are called as the modules or they are communities. Then hierarchy describe how the various structural elements are combined. Then small world network, you can see 2 arbitrary nodes are connected. You can have this here. Then the network has average shortest path length. The paper I have referred here, that is published in 1998. The interaction network that have been shown to be a small network but is connected to another network for significant expression. Now flow chart. If you want to start tutorial mein you will learn it, how to make this through cytoscope and string, data set to be collected from NCBI and GEO database, data normalization, then do the WGCNA, that is a weighted network, connected neural network model, then string, use the string, then you do the algorithm finding and find the hub genes. Hub genes means how they are connected to each other, and if you are able to do that, you will get the network model. Now what is cytoscape and other plugs-in used in the gene network model? So this is for identification of disease. These are the applications, okay. Identification of disease subnetworks, identification of disease networks, subnetworks which are active in disease. When there is a diseased condition and you want to study how disease is going on and how it is happening, so this you can see subnetwork based diagnostics, source of biomarkers, as I told you, for diseases classification, identify the interconnected genes and also see that how these disease prediction can be done. Then subnetwork based genes association, especially the pathway mechanism and the SNPs, okay, single nucleotide proteins. Then coming to the gene network function prediction, you can do the connections using the different proteins which are involved in the network and how they are expressed. Then detection of protein complexes and subnetwork. So as I told you that there are subnetworks also which are playing the role in the final network and nodes. So you can also see about that by seeing the feedback loops and motifs. Then network evolution, how the network is evolved from one level to another level, okay. So here you can understand the biological processes. Then prediction of interactions and functional association also can be done. So pathways, you can see signaling, metabolic, regulatory, whatever pathway you want to do. Then interaction you can study, that is the protein-protein interaction, protein ligand interaction, and domain-domain interaction. These pictures I have presented from my own research paper, which is in draft right now, okay, that is related to the cyanobacterial protein, okay. So we are trying to see that RNA, that is the ribonucleases, how the ribonucleases are important in cyanobacteria and how these ribonucleases are playing the role in the interaction. So in the network, this I already explained. Now you see this picture, this is 200 genes encoding proteins with diverse cellular roles was revealed in non-biased CRISPR screen for regulators of, this is one example, okay. Many of the genes have previously been linked to autism. This is one example given for the disease. Now autism is a big problem these days. So many other problems is there, neuro, again coming to the neuro connection, okay. So if you talk about this problem, there is lot of network which is happening. So you see these are the networks, these are important genes in the RNA metabolism, this is processing, this is DNA binding, then chromatin organization and histone. Any changes will affect the overall expression. So that you have to find it out. So this complex network, how you can study so many nodes and subnets and hubs are there. So you need to prioritize that which gene is involved in the crucial metabolism or related to the autism, taking one example of autism and how they are done. So this is 200 genes which they have encoded for having role in that autism disease. This is a paper of our Professor Azad. They have just constructed this, a gene network driven approach to infer novel pathogenicity, associated genes, application to Pseudomonas aeruginosa PAO1. Okay, so you can see this paper, and they have created gene network pipeline, complete pipeline of this PAO1, and this is a pathogen which has given a lot of clue, especially the functional genomics, and also understand that how this system driven approach can be helpful. And more discussion about this paper and methodology will be done by Professor Azad. So this he recently published, probably last year, and if you see this diagram, how they have made this co-expression network based model, okay, and also they are studying the ARG, that is antibiotic resistant. So this gene, they have studied that, this pathogen, and studied the ARG characteristics, antibiotic resistance. So this is all about this, and I think it's almost done for today, and let me complete the study story, final story. Okay, so this is all about that. Now you will have tutorial. Before that, I will try to complete the study, some of the aspects. I think few of you were absent yesterday evening, so you might have left. Where is my box? कहाँ गया बॉक्स? नहीं, ऊपर तो नहीं रखा है. ओ, आई क्या पे? मैंने रखा. Or we want to cover it later. If you have any questions, we can discuss. That is the right time. Another 15-20 minutes we can do that, because that will have more slides, maybe 20-30 slides more. So it will take another. We can have discussion. That will be better. Okay. Also, I want that few of you can tell whatever research area you are doing and how you use this thing. You want to use this thing and you want some discussion over it. Please, first you.
The slide is shown 200 gene networks. So some circular, small and some circular large. So on what this is diagram.
See, this is taken from one paper of autism. Okay. So here they have seen 200 genes which are responsible for various functions related to this autism. So these diseases are never understood by us. How it happens, why it happens, when it happens, and how these are connected. So in that, the small nodes and bigger nodes, you can see. So bigger circle may have more subnets, okay, subnetworks, okay. So it is only 200 they have highlighted in their paper, okay, and maybe more are there. So you can see the bigger network and a smaller network. So you can connect it with together with the functions, and during tutorial you will learn that how to prioritize which to connect, okay. Like you have, it is simple, like you have I think 30 friends here, you connect with 4, those who are working in your research area. You don't go to the person who is working on cancer biology, you don't go to a person who is working on neurobiology, okay. You connect it with your own network system. So the same function is done in this, that in the bigger area, bigger boxes, there are more nodes available, more input to be required, and some of the crucial things are also shown in the bigger way, highlighted one, okay. And the hub genes are there, you know, the hub genes are also shown in a bigger way. So when you see a bigger circle there, it means there is a hub gene junction, okay. You have lot of information there, put more efforts there, you will get more networks there. It is like that, seeing under microscope again and again and again with different resolutions. So if you have a bigger network available, you can try for it and try to see how this is connected. And it is just a long read aspect. It is not only that you can study it one network, you can study 2 network and 1 method, okay. You have to see that how your expression of genes at various level is connected. If you are targeting a metabolite, if you are targeting a protein, if you are targeting any disease, if you are targeting any type of personalized medicine, if you are targeting any type of application, these are all avenues which you can cover. So first you have to think what you are looking for. Are you looking for a gene which is involved in the increased tolerance of crop to the heat, okay. Are you looking for a gene which is responsible for more xanthophyll production or metabolite production, or looking for a gene which is involved in certain type of autism disease, more evolved there, or you are targeting about a gene which is involved in various type of ailments, disease ailments which are not so common like cancer and all those things, okay, and diabetes also. So these are all things which you have to think about, that what application you are looking for, or you are simply targeting to understand evolution, simple targeting that you have 2 species of microbes in the lab, one you have got from one culture collection, one species already in the lab. Like we have one species of Thermomyces lanuginosus VAPS 25, right, 24 and 25. We got this in 2014. Now it is 25, 10 years ho gaye. Now we have to see, we can see definitely that how this, it is the same network happening, or the simple microbial changes, microbial protein changes are there, and what is happening, what is the capability, how the, is it the same which produces the same type of enzymes. So it is like that. One live example I will tell you. When I went to Southern Ocean in 2011, Antarctica, for sample collection of ocean microflora for a microbiological study. So during the course of that journey, we collected deep ocean samples, and there we collected some deep water samples. From there we got one Bacillus pumilus, okay. That enzyme was, the Bacillus pumilus was producing the enzyme protease, that was alkaline protease. So we started our study, we brought the samples, we brought the samples to the lab. After 1 year, the same Bacillus pumilus was not able to produce the enzyme. So the enzyme was good in 1 year, after 2 years it was not able to produce. Why? The reason being the acclimatizes. The samples which we collected were having different capabilities. So we use sea water, we give all artificial conditions to get that enzyme, but the enzyme capability was not that level. So it is same like that. You need to understand simple, you can study evolution also. That evolution of, like you can think about that how the different species is evolving and what is the network changes happening, what are the nodes changes happening. So you may have same bacillus pumilus, so many bacillus pumilus, so many rhizophosphorai, all they are able to produce the same thing? Not like that. So it is same story with this. Okay. Please. NGS data analysis is based on various softwares which are available. Okay. Normally these days we are very fortunate that the companies, they give you analysis also. So if you talk to them, because the data analysis takes lot of time, and what really you are looking for. Analysis means analysis. What analysis you want? NGS mein what analysis you want? You want the analysis for the pathway, you want analysis for the transcriptome, you want to analysis the whole genome information, or you want to understand the gene level interaction. So NGS information, what information you are looking for has to be analyzed. So you will get information about NGS analysis that okay, this gene is connected to certain type of pathway. That's why you are doing the NGS data analysis. Okay, you want to know that these are the crucial genes involved in this type of function. So based on that you can decide that what analysis you want to do. Okay, the data is there, you can analyze whatever you want. So these services are provided, and I have given you some softwares for NGS analysis. Okay, so you can take up those information. Okay, and before you take NGS services, outsource normally karte hain, so I suggest you to talk to them to provide you the analysis part also. Okay, because they do analysis, they charge a little bit high, but if you ask them, you can do that. And if you are trained, you can have your own startup to analyze and help people. Okay, and get money out of it. Any other? Please, question.
You said that by transcription we can know, by transcriptomics we can know what the response of, like cellular response against the stress and all. So is there any way we can map transcriptomics, like we can map metabolites to transcriptomics? Is there any way?
Transcriptomics to?
Metabolites from the pathways.
No, this is all interconnected. What I told is that you can, if you understand transcriptome, you know how, what type of proteins are there, how they are transcribed, and how transcriptome is designed. So if you are targeting any protein, you know that this protein is responsible for this type of changes in the cell. So you can definitely target that protein and understand that. And also you see that this protein is involved in the metabolomics of the cell. So you can, but you have to identify a specific protein which you want to study. So if it is a big sample, you can't do that. If there are sample numbers are reduced, you have already prioritized by doing the study or previous paper that okay, these are 2 proteins involved in the cell interaction, then you can target. Okay, like sigma factor I told you. In my our research project we have identified 4 sigma factors. All these transcriptional regulators, they are important in the cell function, increased metabolite production. Sigma E, sigma B, C and D. So all these sigma factors we are targeting. We know that these are the sigma factors. First you have to identify what protein you are looking for. Otherwise it is impossible. So we identified that these are the crucial sigma factors involved in the transcriptional engineering for the cyanobacteria. If we are able to control these sigma factors, or we are able to understand their mechanistic working, we can control the cell function. So we want to control, we want to make a strain which is a mutant strain of a cyanobacteria which is a super producer. So making super producer, you should know that. So in the same way which happened, I think 2 years back, some people they did therapeutic cloning and all those things. So you need to understand that can be done, no problem. It is first you identify what protein you are looking for, what function you are looking. Please.
Following this question, so if we use AI and ML, so is it possible that if we have a bigger sample size, it will help us with mapping?
It will be helpful, but sample collection procedure, you have to see that you should have all the parameters. Okay, first you see that what parameters are required to shortlist using AI tools. So you should see that the parameters has to be formulated first as per AI requirement. Then you take the samples. Don't take the sample and then ask AI to do that. First you see what AI requires for helping you. Then you do the sampling accordingly, then you fix in AI tool and get it. Okay, so I have one project, I will tell you. Just 1 minute. We are working into a project, the potential role of AI agents in transforming nuclear medicine research and cancer management. Jyoti, you work on cancer biology, right? So this is a very good thing, and I will tell you, clinical interpretation of image based features and trying to understand whole genome and RNA sequencing, and collection of multi-center validation, that is there. So this is another thing that they do non-invasive techniques like PET-CT, which help us to understand the lung cancer patient. So they do only the PET-CT and use AI tool. Based on that, they will devise a mechanism and tell you that these are the prognosis, this is diagnosis. So this startup is already there, I cannot tell you. The first is the transforming nuclear medicine research. Second is AI agents, role of AI agents in addressing the challenges, and then AI for healthcare. Okay, so they take patient demography, AI agents. If you are interested, I can connect to that. Okay, so lot of thing is there. The plan is already in place. Okay, but अभी उसको बहुत सारा काम बाकी है. And once we are able to do it can be deployed for large scale population also. So AI is very helpful. So many countries they are using it. India is also using it, and they. Any other? If there are no further questions, we can break 15 minutes earlier. Okay, and if you have any other agenda for discussion, we can continue, or otherwise we can close it now. And then next tutorial session we will start around 2:15. Okay, little early, so that you get 15 minutes there more for discussion, 15-30 minutes. Okay, so with this